Welcome, Nancy, to the collaborative research and origin and function of microorganisms. It's very nice that you're here. You give a major talk afterwards. You are professor of integrative biology at the University of Texas, Austin, which is, I really like the name, a professor of integrative biology. That's probably going where the future must be. You're not only well-cited, you're one of the most cited symbiosis researchers which is around. You are one of them, maybe the leading symbiosis researchers, and it's an honor to have you here. If one goes through your, more than 300 papers, there is always a common topic, and the topic is always insects and plant-associated insects and microbial symbionts. That seems to fascinate you from the very beginning of your academic career. Can you tell us a bit, how did you get to that? Was that already as a school kid, or how did you get fascinated to that? For the outsider, still very specific topic.
Yeah, it's interesting. As a little child, I, for some reason, loved insects. I just noticed them a lot and learned about them a little bit and just noticed them around me, growing up. And I didn't connect it at all with a career or even anything that you would study even. And when I first took biology classes, they had nothing to do, like we studied the parts of the ear or whatever, we had nothing to do with the things I was interested in around me. So I didn't really even connect it with an academic approach to anything. But I liked, biology, but still, I didn't really like academic biology because the way it was taught in high school was just terrible. It wasn't interesting, and it was actually kind of repulsive. But then when I went to college, I didn't know my major. I was still not focused on anything, and I happened to take a biology course that had evolution in it, which I also had not heard about in my earlier years growing up. I went to Catholic schools, and they just didn't include that. So I got really interested in that, evolution, and so that is also another theme I've always had. And somehow it came together with insects, and I started studying insects, and I figured out, you could make a career out of this. Some grad students told me, So I decided, okay, I'll go to graduate school, and you can pay your way as you go. And I started studying insects in graduate school, connected to evolution. So back then, there was a big question, the evolution of sex. Why are things sexual? So I started studying aphids, a group of insects that feed on plants, because they are asexual for most of their life cycle. So they were an interesting group for some of those questions. But then I just kept working on aphids, and I had some knowledge of symbionts back then, but this was before genomics, right? I knew that symbionts existed because of this book by Paul Buchner, which was in the library. And, looking in that book, you see, wow, there's this entire world that no one studies. No one studied symbiosis back then. And I was aware of that. And in aphids in particular, they are a major part of aphids. Aphids, a big part of the actual body of the aphid are these symbionts. But no one studied them. They just would speculate sometimes. If there was something they couldn't explain, they'd say the symbionts did it. That was as far as it went. And then I just, once technology changed, I was able to, start studying them, start studying the symbionts themselves. It became possible. So that was kind of it was just one step at a time, realizing, I could do this, I could do that, and following those early interests.
You touched a number of points which we may go a bit deeper. First, I see also parallels to my own career, being driven by an intrinsic interest, but then being in a school which probably is good, but maybe hasn't offered you all what you really wanted, getting in an academic area where you all see, what you can do. And you found your niche, and then you developed that niche to your own career. Now you mentioned, of course, technology and genomics, and now we are in a different world, and you are driving genomics and metagenomic approaches in your field. How important is that, and that goes to big data. I mean, we collect enormous amounts of sequencing data, and you can interfere with interaction pathways and modeling. How does that go, this big data world, with the world which you also like and which you describe as, looking to the organism and trying to deep understand the organism in its biology? Is that a contradiction, or is that just a logical, a new way of going deeper and deeper?
I think it's compatible, that it's a new way of going deeper. I think people can get mired in the data and forget about the bigger components of what they're looking at. So I think it needs to be related back to the actual organisms, like what are the main things controlling the lives of these organisms? You can get a huge amount of data and still not know anything about that if you aren't referring back with some kind of direct information about the organism's direct observations. So I do think sometimes people can go too far just into, sort of burrowing into the data without thinking about how, what's important here in terms of, what really happens with organisms. So we do a lot of somewhat big data analysis, but always have the organisms themselves in the lab or, nearby and experiments going with them as well. So.
Yeah. If you think a moment, what does this new world, this big data world, bioinformatics, modeling, etc., mean for educating a younger researcher? I mean, I find it always very positive if a person is really interested in the biology per se and then has to pick up the new technologies. There's also, of course, technology freaks who are very driven by mathematics and biostatistics, etc., and sometimes forget the biology. Is that a challenge for educating the new generation of symbiosis researchers?
I think it's something of a challenge. People get motivated by different things originally. Some people, it's the bugs themselves, so they're interested in them and they want to learn the tools to understand them. Other people just love technology and, coding and data and dealing with it and actually are not initially interested. And so I think that's fine, and both sides can contribute a lot, but the communication is kind of the barrier. They have to communicate,
At the end, if you want to become a established researcher like yourself, I mean, I guess you have to combine both sides. I mean, thinking big, but also thinking small. And yeah. Brings me to another point, what we just talked about, and that is, I mean, I know you a bit, and I know you love to hike and go in the deserts and spend your vacations there and look for insects and look into the nature. That's wonderful. But there's our science and the world, the competitive world of even host-microbe interaction is still dominated by model organisms. And if you want to, publish high, you have to have an established model system where tons of data are already available and you contribute. Now, you are a wonderful example where you go out, you find an interesting insect, which is just a very peculiar ecosystem inhabitant, and you take it maybe to the lab and do research. Then people would call it as a non-model. How important are non-models versus models?
Well, non-models are, most of the world, right? So, we need to be interested in what's actually out there. It turns out that I think the most important model organisms in biology, they tend to be rather unusual in not interacting a lot with microbes. I mean, Drosophila melanogaster and even C. elegans, and not having at least very big conspicuous symbiotic relationships. And that makes sense because they're things you can grow quickly in the lab, without a lot of the other biological input that most things living out in nature would have. A lot of things living out in nature are hard to grow in the lab because they depend on so many other organisms and interactions that it's actually hard to isolate them in the lab and study them like that. So, of course, it's a huge advantage to have something that you can grow quickly and simply and understand the basics of genetics and all kinds of things that way. That's a lot of progress. I mean, enormous progress is made that way. But then you need to look around and say, well, what do we need to explain that we're not explaining with these models? And what are there some at least sub-model, sort of simple models where we don't have all the tools, but we at least can have different investigators studying the same species and looking at their interactions. And so like the aphid model and the honeybee model, I've tried to help develop them to a point that multiple people can study them, can study the same strains. It can, basically try to look at, kind of join forces together, different laboratories, and answer questions. And even though they're not at the level of Drosophila melanogaster or yeast or something, in terms of the tools and the genetics, still, they're somewhat developed systems where people can take it a step further and then a step further, as opposed to just starting over.
Means it's possible to become a model from a non-model.
Right, that's how they all started.
And that's how they all started. And, even honeybee today, but as you say, you developed that. The aphid model, you developed that. And over the years, you also, uncovered many of the interactions between the host, the plant host, the insect, the symbionts in the insects. I mean, this is an unlimited complex. In 2018, you had a paper in PNAS with the title "Glyphosate Perturbs the Gut Microbiome of the Honeybee." And I think this year you continue on that story and on that problem. This is very worrisome, of course, that this happens, and you pinpointed down in the honeybee gut what's really happening. Why is it worrying you, and is there anything what we can do or what you can do against that, using of these pesticides and chemicals?
Yeah, I think in the case of glyphosate, in a way it was actually what we found was expected, because the known target very well, demonstrated that is an enzyme that's present in plants, bacteria, fungi, is present in everything except animals, right? So glyphosate should be non-toxic to animals, but if animals are depending on a microbiome that has this target enzyme, then we actually expect and would predict, and that's what we found with the honeybees, so that their gut microbes are susceptible to this molecule, and that it has a big effect, and it increases mortality of the bees. The gut microbe in the bee gut are beneficial to the bees. They protect against other pathogens and so on. So the perturbation of the gut microbiome actually leads to higher mortality. So, so it's all, I think it's worth knowing this. I mean, it is worrisome. But just the knowledge of it, at least we know we shouldn't spray the beehives themselves, right? So, which it wasn't known because it was just considered to have no effect. So we at least know that if glyphosate is going to be used, then it should be used to cause the least harm possible. Mm-hmm. But there's also the question of all the other microbes in the soil, on the plant surfaces, and all of those, which we generally expect to be affected by that particular pesticide, herbicide.
We have tons of microbes in our body, and we also depend on them as the honeybee. So isn't that also pointing to a problem for humans?
Could be. It's all a matter of concentrations and whether it, how it's metabolized in a given system. Mm-hmm. But it seems at least something that could be happening. We haven't studied human or mammalian guts, so.
Yeah. I think it was for me it was an awakening moment to get this paper, and because there's at least this honeybee gut, there's a solid data which clearly show what's going on. So I think it's very important. Yeah.
It was a grad student who did most of the work on that, Eric Mota, and he after that paper he followed it up with more studies to just confirm and also look more at the dose dependence of the concentration, and all of it's been really consistent with particular microbe that we consider sort of a keystone species in the microbial community is particularly knocked back again and again. And actually, other labs other than ours have also done studies and shown this. So that's a definite, repeated result. And so I thought after that paper, he should do further studies to make sure there's confidence, and it wasn't just a one-time thing. So
coming back a bit to technology and also maybe to the future. I mean, if our modern life has the consequence that obviously we lose diversity of microbes, or maybe we lose particularly important bacteria, and we and many animals. You also showed in a paper that by genetically engineering bacteria, you may show a way how in future one could maybe approach these problems. Could you explain a bit the thinking behind that approach, and also is that one of the ways of how we should maybe respond to these modern challenges of our time, lifestyle and climate change and everything changes? I mean, are there now technological ways of how to respond to that?
Right. So first, I'll just say a little what we did. We took a major bacterial symbiont that lives in the honeybee gut and genetically engineered it. This was all done in the laboratory, and in a way that it would express RNA that would double-stranded RNA that would trigger the bee's own immune system, called the RNAi pathway, would trigger that system to kill the virus, and it worked. We even managed to do it in a way to kill the varroa mite that's feeding on the bee, and so these are possible approaches that could modify, could be used to control pests. They could be used to increase survivorship of beneficial, pollinators and so on, and I know so far these are not approved for use outdoors, and because people have these concerns about genetically modified anything. But on the other hand, what we're doing right now is pouring chemicals on everything. It's so much more damaging than a little piece of DNA. That DNA is flying everywhere all the time, and it's not generally harmful. But the chemicals that we're pouring on the landscape, and for example, in bees, a lot of chemicals are put on hives to control the mites. And there's a lot of interest now in controlling crop pests by using these kind of approaches, genetically modified bacteria that are living on the surface of the plant that basically kill. And these are beneficial or desirable, I would say, because they're very targeted to that organism. The sequence matches, it kills that, nothing else, as opposed to these generalized toxins that kill everything around. And plus, if one problem with insecticides, for example, is evolution gets ahead of them, right? So the insects evolve resistance, and then you have to make a new one. With these, probably they can't evolve resistance given the details of it, but if they did, you just change the sequence to match it, and it would be a very simple fix. And it would, again, be very targeted to that particular pest. So there's a lot of interest in those approaches for crop pest control. And I think it's better than what we're doing now, which is really harmful. Killing all insects in a landscape is harmful. So I think there's also interest in the use of this kind of thing for therapeutics, for medical use.
Genetic engineered, yeah.
Yeah, a genetically engineered bacterium that could colonize the gut, that could maybe some metabolic deficiency that a patient had, or could specifically kill a given pathogen that's in a patient. And those are going to be a lot better than some of the therapies that are used that are very harsh on the whole body and so on. So it's a matter of getting them working well and then having people understand it, basically, so that they can see that it's a solution.
So, yeah, I fully agree, and I think this is one of the modern genetic tools of maybe getting very target-specific changes in the desired manner. If you think a bit broader on host-microbe, host-symbiont interactions, What are still challenges in the field? What are black boxes which need to be addressed now? Maybe which only can be addressed now with novel technologies?
Right, so we now know that, symbionts are really important. It's actually pretty easy to show that, the hosts without the symbiont and with the symbiont are different, and that it's generally better to have symbionts than normal symbionts. And that's a fairly easy observation, and it's very general. We see it in plants, animals, everything, So we see that, but we don't know how it works in most cases. What are the mechanisms, exactly what is the microbe doing that affects what process in the host? Very rarely do we know those things, and people are, pursuing those now, but still is, for most systems, it's still a black box. And is it going to be a lot of different mechanisms, different for every system? Probably not, because why would there be such a broad similarity in the observation? Probably there's some commonalities, at least somewhat, across systems. And we know that a little bit already, but that, for example, short peptides are important in these interactions, both for plants and for animals. Those are, and so that some generalities like that will probably come out. But the new technologies where you can look, at given molecules, you can do microscopy and actually see where given molecules are and see who's producing it, who's receiving it, and so on. Those will make a big difference for figuring these things out. But there's really a lot we don't know. We know they're important, but the details are still missing.
That's so wonderful to hear from an established senior researcher, And I think they're absolutely the same. There is so much which we do not know. That's the future for the young generation. We are coming to the end, but this is, I think, a good point of thinking on the younger generation. You are established, you are a senior. For many, you are a hero in the field. In your opinion, and you showed us told us at the beginning how your own pathway was from childhood to your career. What would you say to a young an early career scientist that, as we say now, male or female or male, what does it need to become successful in our field? Is there anything which is really necessary, or is all by chance, or would you give an advice to them? What does it need to become successful at the end?
I think being interested in a variety of things and trying to see how they fit together. So not just going deeply into one little field where people are doing the same thing and trying to do it slightly better than they do it. Because usually the advances come from connecting things from different fields. And so the first thing I would say to this young person is, what are your real interests? What really excites you? Make sure that you include that in your plans, because sometimes people worry so much that they don't remember why they even are doing it. And then to kind of be open to reading different things and looking at, different systems, or, going to talks where you hear different points of view, because then you can put something together and you have a chance of doing something new, as opposed to just slightly tweaking what other people have done who are working on one given system. And at least for me, that's a lot more fun than slightly modifying, someone's protocol for some particular procedure. So being kind of open and obviously you have to be curious to start with, otherwise it won't be fun and you won't want to keep doing it. But if you're interested in something, there's probably many different ways to approach it, and you should kind of try to sample those things as you run into them, that people might be using it on some other system, but you could see how you could use it here.
Did you ever worry in your life you don't get there where you want to get to, or that was never any issue?
I never really worried. I actually, one thing I notice, a lot of younger scientists seem to worry a lot. They all seem worried, like an epidemic of worrying, and I didn't, because I just figured out the next step. I guess before I got my assistant professor job, I wanted to get one, right? So I guess I was a little worried about that, but I don't know. I don't think I worried at the level that many people do nowadays. And if anything, I had more reason to worry because things were less predictable. I think now the systems are more predictable. Of course, it's hard to get permanent position pretty much in every country, but it was hard then too. And if you look at the numbers, it wasn't really very different. So I think the main thing is to keep doing things you like. There'll probably be a way to keep doing those, and by chance you may end up here, you may end up there, but you'll still have done something that you like. So
I think this is a very good advice for the very end. Don't worry too much, but keep going what you're interested in doing. I think this is the point. Yeah. Thank you so much for sharing your thoughts.
Thanks.
And for being with us. We are looking forward to your talk. Thank you very much for coming.
Great, thanks a lot. That was fun.